Hey guys, my name is Brooke Martin, and today I'm going to talk to you about common problems seen in the Molecular Parasitology Lab. In this lab, we work with Crithidia fasciculata. The purpose of the lab is to determine the function and localization of open reading frames in the genome of the Crithidia. We determine localization through isolating the open reading frame, ligating to a plasmid, confirming ligation, transfection, and then microscopy. To determine the localization, we tag our open reading frames with green fluorescing protein, also known as GFP. In the class, we use the penis plasmid to achieve tagging our open reading frame with GFP. It is important to put cells directly into the ice after retrieving them from the negative 80 freezer. This will prevent any damage to the cells. The heat shock introduces holes into the membrane of the cells so that the plasmid can enter. After the heat shock, it is important to let the cells recover on ice. After incubating on ice for 2 minutes, we add superoptimal broth or SOC to the cells. Adding SOC should be done under flame to ensure that no contamination occurs. SOC should be incubated and checked for growth the following day to determine if contamination occurred. After cells incubate in SOC for an hour, you spread on nutrient agar plates. In this lab, we commonly use LB ampicillin plates. Aseptic techniques must be used to ensure no contamination occurs. When spreading, we use a sterile metal spreader. It is important that it is metal so that when you flame it, it gets hot enough to kill any lingering bacteria. During transformation, if you're using new products such as plates or cells, it is important to make sure that they are viable. To do this, we use positive and negative controls along with our samples. A positive control would be cells without a plasmid on nutrient plates without a selective antibiotic. A negative control would be cells without a plasmid on nutrient plates with antibiotics. We would expect to see growth on the positive controls and our samples. We would expect to see no growth on our negative control. There are two main types of bad results seen in transformation. The absence of growth on all of the plates can occur for several reasons. The plates could be made incorrectly, the spreader was not cooled, or the cells were not maintained properly and died. When this happens, go over your protocol and be sure you are not missing any steps. Growth on all of the plates is a sign that the plates were made incorrectly, or that the cells you are working with naturally contain the antibiotic resistance that you were selecting for. When this happens, go over the protocol with the individual who made the plates. Also, check to see what strain of bacteria you are working with. The next step in the sequence of methods is to determine if our open reading frame was successfully incorporated into the plasmid through restriction digest. However, we also use restriction digest to isolate our open reading frame. In this lab, we use the enzymes EcoR1, KPN1, and NEAD1. They contain the following restriction sites, and their protocols can be found on the following QR codes. First, we digest our open reading frame that was previously incorporated into PJET 1.2 to isolate the gene. Many of the open reading frames use KPN or need one, but Bagel can be used too. This depends on previous semester's work, so be sure to check their sequencing data before starting. After digesting our open reading frame with the proper enzymes for isolation, we digest circularized penis with the same enzymes. This yields a linearized plasmid that our open reading frame can be incorporated into. Then we ligate our open reading frame to penis with T4 DNA ligase. Transform, then digest with EchoR1 to determine if our ORF was tagged with GFP. You must gently flick the solution to mix them. Reagents that are not mixed properly could introduce a higher or lower concentration in the reaction, which could affect the sample. Next, you must do a short spin on the centrifuge. This ensures that all of the sample is in the bottom of the tube. This is particularly important when working with very small volumes of solution. There are many different bad results that can happen in restriction digest. A common mistake is seeing bands that are larger than the plasmid. In this example, we are looking at open reading frame 8996 attempting to be ligated to penis GFP. In this gel, we see two bands. One is over 20,000 base pairs and the other is around 5,000. Because open reading frame 8996 is 573 base pairs and penis GFP is 7,190 base pairs, we would expect to see a plasmid around 7,763 base pairs. The lower band in this example is likely the plasmid in a conformation that makes it appear smaller than it is. The higher band is likely contamination in the sample that is genomic DNA of the E. coli. To avoid this, be sure to follow the plasmid prep kit instructions precisely. Another common mistake is not handling the enzymes properly. This will denature the enzyme and restriction sites will not be cut. In this example, open reading frame 9990, which is 348 base pairs, is ligated to penis GFP, which is 7,190 base pairs. When we cut the plasmid with EchoR1, we expect to cut out the open reading frame tagged to the GFP. This band should be about 1,079 base pairs. Even if the open reading frame is not incorporated into the plasmid, we would expect to see GFP cut out in a band that it appears around 731 base pairs. In this example, this is not the case, which means no enzyme was added or it was denatured. The next example may look like it is a mistake. Two bands appear in the bagel and double digest lanes. At first glance, it may look like an additional enzyme was added to the bagel control. However, after carefully reviewing sequencing results, it appears that PJET plasmid contains two restriction sites for bagel. This misinterpretation could cause perfectly good results to be thrown into the trash. Be sure to check the plasmids for what restriction sites they have before starting. In this example, bands appear at an undesirable size. Open reading frame 8996 was to be ligated into the penis GFP plasmid. Digestion with EchoR1 will release a band about 1,304 base pairs because open reading frame 8996 is 573 base pairs and GFP is 731. Unfortunately, only a band of about 731 base pairs is released, which means the open reading frame was not incorporated into the plasmid.